Hello everyone, in continuation with the previous video of asexual reproduction, we will be talking about regeneration, budding and spore formation in this video. What is the meaning of regeneration? Regeneration means it's generating again. Organisms like hydra, planaria and starfish can be cut into any number of pieces, especially starfish at specific locations, but other than that, planaria and hydra can be cut into any number of pieces, and each piece will grow back into a complete organism. This is known as regeneration. So as you can see here in this picture, planaria, it's cut at the head part, which can grow back, the tail part, which can again grow back, and the middle part. So the planaria is cut into three parts: first, 2, 3. So all the three parts can regenerate back into a full organism. So regeneration is carried out by specialized cells. These cells proliferate. Proliferate means they increase in number by continuous multiplication and division. So they keep multiplying in number of times, and a large mass of number of cells are formed. A large mass of cells are formed, and this mass of cell will arrange or will group again into different forms, and different cells undergo changes. The group of cells will undergo changes to become various cell types and tissues. So we'll quickly have a look into the regeneration of the planaria, and then we'll come back to the regeneration of starfish. So as you can see here, the day 0 amputation means we are going to cut the planaria with the help of a knife into 2 pieces. Can you see this planaria? They have been cutting it into 2 pieces. So that's the head part, and this is the tail part. This is the tail part and this is the head part. As you can see here, both the parts are moving individually, correct? And now we look into what happens with the head part and how it develops back into the whole organism, and then we look into this. So this is the tail part, okay, the first day. So gradually it is developing. Can you see this? Gradually it is increasing in size and it's going to develop. So the head part is coming out like this. The transparent section will be developing into the head part, and gradually as the number of days increases, can you see the head part here, right? And it will develop in such a way that you can identify these two will become the eye of the new organism. Can you see? These are the eyes. And it's going to develop into a whole new organism. Now these eyes are really visible. Can you see that? Now the nose part is to be developed. Nose-like structure has to be developed, and then it will completely change into a new organism. Can you see the eyes, distinct eyes? And then the nose. This is how the final tail part develops into the whole organism within 4 days. Now we are going to talk about the head part, which was cut into half. So the head part is already having the eyes and the tail, and the tail part has to be developed. So gradually this tail part will start. The part from where it is being cut off, it will be developing into a tail. So can you see the tiny bit of a tail which is being growing? So you can clearly see the tail part being developing. This transparent section is the one which is going to develop into the tail. Can you see? And the tail is completely developed. Again, the head part will develop into a complete organism within the 4 days after the amputation. So as we saw in the experiment, the head, tail and the middle part, they individually can grow back as a whole organism. So this is called as regeneration. Now the changes that take place in an organized sequence is referred to as development. Regeneration is not the same as reproduction because many organisms will not be favoring this method of reproduction where they are being cut into pieces and they develop into a new organism. So in 14 days, a planaria will grow back to its whole structure. Not 4 days, I'm sorry, it takes 14 days. So whenever it is cut into pieces, each piece will develop back into a whole organism in 14 days. So let's look into the starfish. As you can see here, the starfish is having 2 tentacles. The remaining 2 parts are being developed. Can you see the 2 sections are there? Normally the star has 5. The 2 are there. You can see the tentacles moving, but at the same time the 2 new are being developing here. This part, the bud-like structure which is coming out, which will develop into the pieces. These 2, right? So in true sense, regeneration is not considered to be a type of asexual reproduction because it's not a natural process. Some external factors have to work on it to break them into pieces, and then they'll regenerate or they'll develop back into an organism. Next type of reproduction that we are looking into is budding. So this is a hydra. Okay, this is the bud that you can see, right? So what does hydra take up as method of regeneration? So hydra uses regenerative cells. Regenerative cells means those cells which can generate back, reproduce back, okay, in the process of budding. A bud develops as an outgrowth due to the repeated cell division at one specific site. It can't do any other place, but only that one specific site where it can develop a bud, like in this case, right? These buds develop into tiny individuals, and once they are fully mature, they detach from the parent body and become new independent individuals. Let's have a look into the bud of the hydra. As you can see here, this is the main hydra and this is the bud of the hydra. And that bud is specifically growing at a specific location, as you can see here, right? And it is attached till it doesn't get matured. It is still attached to the main hydra, that is the parent organism. So the daughter's organism is still stuck to the parent unless and until it doesn't get matured. It will keep attached with the parent, and once it is matured, it will get detached and grows back. The next type of budding which we can see is in yeast. So yeast also reproduces by budding. First of all, in yeast, what happens is the bud appears on the outside of the cell wall. As you can see here, this tiny bud here, and the same with this tiny bud here. Once the tiny bud is appears at the outside of the cell wall, the nucleus of the parent yeast cell will then divide into 2 parts. As you can see here, there are 2 nucleuses, right? And then what happens? One of the nucleus will move into the bud. And ultimately, the bud will separate off from the parent yeast cell and forms a new yeast cell. So let's look into this process also. As you can see here, this is the tiny bud. The nucleus has moved from the parent to the bud and it's gradually growing. Similarly, the other buds are also growing, right? Can you see it? The tiny 3 buds, the nucleus gradually moves into them and then they start growing bigger and bigger and bigger. So thus, once they are mature, they'll get dead. So we have seen 2 types of reproduction. One was regeneration, which was in the planaria and starfish. The budding process we saw in hydra and yeast. The third type of reproduction that we talk about is spore formation. What is spore formation? Now, spore formation, it's actually in many simple multicellular organisms, there are very specific reproduction parts that can be easily identified that these are the parts which will further develop, help in reproduction process or help in producing a new organism. But in rhizopus, the thread-like structure that develops on a bread mold, when a bread is getting spoiled, a thread-like structure forms on it, which we call as a bread mold. But that thread-like structure is not the reproductive part of rhizopus. Instead, the tiny blob-like structure, which is on the stick, like these tiny blob-like structure that you can see on the stick, are the reproductive part of the rhizopus. So now what happens? Normally the thread-like structure looks like this. Gradually, when we magnify it further, we can see the blob-like structure. The blob-like structure has tiny spores in it, which are these tiny structures. These spores are being tightly and intact packed inside the blob-like structure called as spores, and each tiny structure is called as sporangia. Okay, so these sporangia keep developing, and they are intact into the spore, and they keep developing and maturing when the right surface of moist surface is available to them, or when they come in contact with the moist surface, then the spore will rupture and these sporangias will be spread out, and then these sporangias will develop into new organisms. Okay, so these blobs are sporangia which contain cells or spores that can eventually develop into a new ribose of individuals, which you can see here in this picture. The spores are covered by thick wall to protect them from any external factors. They are protected until they come into contact with some another moist surface. Once it happens, the spores will rupture off, and these sporangias are spread out in the nearby contact space, and the sporangias will develop into new rhizopus individuals. So this is how spore formation helps in reproduction, asexual reproduction method of developing a new organism, and it is found in rhizopus bacterium. Hope it is clear. Again, the three types of reproduction that we saw was regeneration, spore formation, and budding. Thank you.